Hello, everyone. I'm Dr. Terry Constantine. I'm the CEO of Brain Research Foundation. And it's my pleasure to introduce Dr. Mitch Roitman from the University of Illinois at Chicago. Dr. Roitman is a past BRF grant recipient, and he's going to tell us about his research, looking at the links between thirst, hunger, and addiction. We will have some time at the end of the talk for questions, so please type your questions in the chat. That's at the bottom of your screen. Now, let me turn it over to Dr. Roitman.
Hello, everyone. Hello, Terry. Thank you, Terry, for that introduction. And I think by way of a little bit of a further introduction, I'll start with a broad overview of my research interests. And I want to begin with a very simple statement, and that is that the value of our goals is dynamic and subject to change. Case in point, this now old picture of my son, Benjamin, who's reaching for this highly rewarding board book, but he's getting an aversive consequence, this stiff arm to the face from his sister Isabel, twin sister Isabel. And you can see that that's evoking a negative emotional response in Benjamin. And that reaching behavior that you see Benjamin exhibiting here, if this had been a video, the reaching behavior would have rapidly changed to avoidance behavior. And so Benjamin learned a lot from this experience. He seemed to shy away from this board book in the future. And he also learned that his twin sister is not someone to be trifled with. So the value of our goals can change as a function of learning, like what you see going on here. But they can also change based on our physiological state, like how hungry or sated we are. As another example, and this one is slightly painful for me because it's been since breakfast that I've last eaten. So for me, this picture of these lovely looking sliders here evokes a lot of hunger, evokes a lot of interest in the burgers. But my response to this picture might be very different if I had just finished dinner, just polished off a plate of these sliders. So the way we value food, nutritive stimuli, can change as a function of our physiological state, hunger or satiety. But it turns out that the same is true of non-nutritive stimuli as well, including drugs of abuse like cocaine. And we've known for decades that hungry subjects respond a lot more vigorously to drugs of abuse like cocaine. In addition, hunger can drive relapse to drug seeking in animals and humans that are normally abstinent, that have recovered from a period of drug taking. So again, by way of broad overview of my research, I'm interested in trying to understand the signals and brain circuits involved in approach and avoidance behavior, and trying to understand how these signals change as a function of learning or changes in our physiological state. Now, I'm a behavioral neuroscientist. So as behavioral neuroscientists, we're constantly trying to establish brain behavior relationships. And probably for the past century, we basically ask the same three questions. Is a brain region necessary for a given behavior? Does activity in that brain region correlate with a given behavior? And finally, is activity in that brain region sufficient for a given behavior? These questions that we ask haven't really changed much in essentially a century. So what has changed? Science obviously progresses. And the idea that the change really has been the development of tools to ask these questions in better and better ways, to sort of develop new knowledge, to develop the precision treatments, precision therapeutics for disorders of brain function, like those that underlie overeating, which can lead to obesity, drug taking that can lead to drug addiction. And so innovation in research is absolutely essential to the development of precision therapeutics. And I'll give you another example of just how important innovation is, and I'll use a little bit more of a personal example. and that is that my father loves opera. And since my earliest days, I was woken up on the weekends to incredibly loud opera music. And in my very early youth, he would listen to opera through records on the record player. But as I aged and as technology got a little bit better, he transitioned to building his opera collection on reel-to-reel tape. And I would ask him, well, hey, what's wrong with the records? And he would tell me that by playing the opera on reel-to-reel, he could hear things that he couldn't hear in the record. The sound was crisper. He could hear the horns better. Of course, reel-to-reel gave way to newer technology, better technology, like compact discs. And again, my father would say how much better the clarity of the sound is with compact discs. And of course, today, he doesn't even bother with collecting physical recordings of operas. They're all as files on his computer. And the technical limit is in the quality of his speakers and not in the device that plays the recordings. I think that the evolution of science is very similar. Especially within behavioral neuroscience. So in early days, maybe a couple decades ago, our measurements, our technology, our tools for establishing brain behavior relationships were relatively crude. In this example here, this is a side view of a rat brain, and here the scientists made basically a cut in the brain to try and isolate the back of the brain from the front of the brain. Over the years, the techniques have improved. This is an example of a more specific, a more selective lesion of the brain to again try to establish brain behavior relationships. We get a little closer to today, and our tools have developed dramatically. Now we can target specific types of neurons and activate them or shut them off at precise times. And that has really allowed us to make a big leap forward in establishing brain behavior relationships. And to figure out what the techniques, what the tools are of really today and tomorrow, I actually make use of science Twitter quite a bit. And I find that I'm able to keep up with innovation through social media. But innovation, that's really the key term that I want to focus on for the next couple of slides here. And I think innovation is really where the BRF comes in. And so Terry mentioned that I'm a recipient of a seed grant. I think the BRF funding occupies a unique space for the evolving scientist. It's really where BRF support helps scientists make the leap from the technologies of yesterday to the technologies of today and tomorrow. And let me give you a little bit more color for that statement. Scientists ultimately need grant funding from the NIH to be successful. I have NIH grants. I need them to fund the bulk of my research program. When I write a grant to get funding from the NIH, there's a section that I have to complete called innovation, where I sort of tell NIH all the new things that I'm going to use, all the new things that I'm going to discover. But really, there's a disconnect between what the NIH says it wants and what it ultimately funds. And I think of this as kind of a seesaw between innovation and feasibility. So when the NIH says it values innovation, I think grant scores actually reflect risk aversion and the valuing of feasibility over innovation. What do I mean by that? They basically want to see that you can not only do the experiments, but that the experiments are going to pan out before you ever get the funding to do the experiment. To that
Mitch, you are breaking
the
up.
experiments are going to work to show your hypotheses before you ever get the funds to do the experiments. And in my opinion, this is really where the BRF steps in. So I think BRF seed grants in particular play a crucial role in giving scientists the space to be innovative. And it says it right on the website, and this is a quote directly from the website: "We work to fund the most inventive, high-impact research projects." I think the BRF is what allows scientists to truly be innovative. Before knowing the answer, develop tools, develop your questions to bridge that gap between adopting the tools of today and tomorrow and reaching out to NIH for further funding. So let me give you a more zoomed in look at my research and how specifically the BRF helped me. I told you at the beginning that I'm broadly interested in understanding motivated behavior. What pushes us to seek food? What pushes us to seek drugs of abuse? And over the decades, over the centuries, we've zoomed in on a particular area of the brain that we think is important. And it's called the ventral tegmental area. That's the little blue dot on this cartoon of a human brain on the left here. But the ventral tegmental area is a very complicated region of the brain. It's made up of lots of different types of individual brain cells called neurons. And we can kind of think of the ventral tegmental area as a bag of M&Ms. We've got orange M&Ms, we've got yellow M&Ms, we've got the brown ones, all different types of neurons within the ventral tegmental area. And so I and many people in the field, when we think about goal-directed, motivated behavior, food seeking, drug seeking, we hypothesize that it's actually the dopamine neurons that are within the ventral tegmental area that are the key elements for motivated behavior. The question is, how can we study this region of the brain and isolate, focus in on what we think are the key neurons for the behavior that we're interested in? That is, how do we measure, how do we manipulate just the green M&Ms and not the other M&Ms in the ventral tegmental area? And for me, this is where the BRF stepped in. This is where the BRF helped me be innovative in my science. And the way that they helped me was that I was able to have a little bit of money, a lot of money, I should say, and space and time to pair the use of transgenic animals. Don't sweat the details here. But the way that we can use these animals is to exclusively express constructs that we place into the ventral tegmental area, place into the dopamine neurons, and allow them to be expressed only in dopamine neurons. And you can see in the image on the left here, what's being expressed only in the dopamine neurons happens to be a fluorescent molecule. And the idea here is that when the dopamine neurons become active, they glow. And I can actually put a fiber optic in the brain of these animals and measure the fluorescence as a proxy for the activity of dopamine neurons. And I'll show you what that all looks like with this little video here. The red line shows you that the time at which a cue was played to our animals. The cue means that the animal is about to get a food reward. And what you'll see scrolling along here is the activity of dopamine neurons that I was able to record only through the help of the BRF, only through the adoption of this relatively new tool. So here goes the video. And so you'll see just a little bit of activity. Here comes the cue, boom, big response in dopamine activity. And there'll be two more trials right on top of each other. You'll see them superimposed. This is an entirely separate trial. We play the cue, boom, big increase in dopamine. So that dopamine response to the cue for me, is kind of jaw-dropping. We're able to ask the dopamine neurons what they're doing in real time, and they clearly respond to specifically the moment that this cue turns on. And I told you in the video that the cue tells the animal you're going to be able to get access to food. It doesn't matter what the reward is. You see the exact same response if you train the animal to expect, instead of food, drugs of abuse, like cocaine. You see the same dopamine response to a cue. And we think that this dopamine response evoked by the cue is important in pushing the animal to approach, in pushing the animal to consume. And of course, when it comes to food and drugs of abuse, too much approach, and you develop things like obesity, things like addiction. So in my lab, if we turn back to the questions that we're asking going forward, we're trying to figure out ways for us to turn down the volume of this brief dopamine spike evoked by the cue. And we have a hypothesis in my lab that we could use the body's own hunger and satiety signals, and I referenced that all the way in the beginning of the talk. We're asking whether we can use the body's own hunger and satiety signals to change the volume, to lower the volume of these dopamine spikes, not just to food, but those that predict drugs of abuse as well. And so here's the only little bit of data that I want to share with you this evening. We're focusing on a signal in our gut called GLP-1. It's released when our gut senses nutrients, senses food. So when I'm done here, I'm going to go have dinner. I'm going to eat a lot. And as my stomach stretches, as the food reaches my intestines, GLP-1 is going to start to be released. And the idea is that that hormone then feeds back on my brain to say, hey, you've had enough, slow down, stop eating. And it turns out that we, the scientific community, the medical community, we're already using drugs that mimic this hormone for the treatment of things like type 2 diabetes and obesity. Liraglutide is a drug that's already FDA approved on the market, and people are using to control their diabetes, to control their obesity. And so we've been asking whether we can use similar drugs that act on this hormone, that act on the hormone's receptors, to tamp down, to lower the volume of those dopamine spikes to drugs of abuse. And so here's just a little bit of data to illustrate that we're well on our way. In the open circles, you can see the dopamine signaling. This is a summed graph, so forgive the brief treatment here. But in the open circles, this is dopamine signaling as animals are getting infusions of cocaine. And in the control condition, the open circles, you can see the dopamine signaling goes up and up and up and up all across these brief hits of cocaine. But in blue, in the dark blue, and at the time of the vertical dashed line, we administered this drug that acts at GLP-1 receptors. And you can see that that very nicely tamps down the dopamine response that would normally be there to drugs of abuse like cocaine. And that is that we think we're starting to lower the volume on dopamine signaling as a means to help individuals prevent relapse to drug seeking when they've managed to become abstinent. So I know that there are many of you on the board of the BRF, and so before I finish up and take your questions, I really want to take a moment to thank you for supporting such a critical foundation. And really what you're doing with that support is truly supporting scientific innovation. And so, as Terry mentioned, I received seed grant funding in 2012. And since that time, I've been awarded multiple NIH grants, almost $3 million in NIH funding, and that's the direct costs. Add 57% to that, and that's the cut that my university takes from the grants that I'm awarded. But in addition, we published 19 peer-reviewed manuscripts based on the work that's flowed from the seed grant funding from BRF. And I've been through all of this time since 2012. I've been able to support multiple postdocs, graduate students, and undergrads, training the next generation of scientists, which is something I value tremendously. So I want to take your questions. I want to hear your thoughts, but I also want to acknowledge my partners aside from the BRF that have helped me to reach this level of insight. These are just a couple pictures of my lab over the years pre-COVID, which is why we could be all cozy in these pictures. And here are the folks that are currently working with me, our lab alumni, a couple of key collaborators, and I always acknowledge the BRF for its seed grant funding, which has really helped launch much of the current research I'm doing in the lab. So with that, I'm going to keep quiet for a few moments and hopefully hear your thoughts and questions.
Well, first, I want to say thank you so much, Mitch. That was pretty interesting. I enjoyed it. I have some follow-up questions, but I don't want to barge in if others do. Since we're a small group, I think that if we can unmute and mute back if we want to ask a question instead of chatting, or whichever you prefer. But let me kick things off. So, you're looking at dopamine. I always think about dopamine, and then I think about Parkinson's disease, and then I think about movement. But this isn't movement. So dopamine is involved in rewards. That's what you're saying as well. So reward response, and that's why you chose it?
That's right. So really, if I'm giving you the quick answer, we basically have two clusters of dopamine neurons in the middle of our brain. So the blue dot I said was the ventral tegmental area. Mm-hmm. And the quick answer is that we basically have another cluster of dopamine neurons called the substantia nigra. And those dopamine neurons send their talk to a region of the brain called the striatum. And that pathway, the substantia nigra to the striatum, that's the pathway that gets destroyed. That's the pathway that degenerates in Parkinson's disease and is critical for movement. The ventral tegmental area, this other cluster of dopamine neurons that I'm interested in, they talk to a different area of the brain called the nucleus accumbens. And that pathway is very important in reward and reinforcement. Now, you said to me that isn't movement, but actually it is, because when you think about what reward and reinforcement is, it's basically saying, hey, whatever you just did, that bite of burger, that snort of cocaine, whatever you just did worked out pretty well, do it again. And that's really what the ventral tegmental area is about. It's kind of movement, but in the context of reward and reinforcement.
Got it. It's a reaction, right? It's a behavior reaction. Yeah. Okay, now I get it. Are there any questions? See, maybe another chat. Are there medicines that have been approved, like the one you're highlighted for type 2 diabetes, that can work to stop humans from wanting alcohol or drugs? If no, what stage of research are the trials? Or trials do you think these medicines might be today? That's a great answer. Question. I was wondering that myself.
It is a great question. And I'm not alone in the interest in our body's own hunger and satiety signals for the treatment of addiction. I think my lab focuses specifically on how they impact dopamine, but actually, let's focus on GLP-1 for just a moment. And it turns out that when people have surgery for bariatric surgery, in cases of severe obesity, they lose quite a bit of this GLP-1 signaling. And what can happen is alcoholism can actually develop as a function of losing this GLP-1. There are groups that are focusing on translating this medicine that's already FDA approved for the treatment of type 2 diabetes, the treatment of obesity. There are groups that are already working on translating that to the treatment of alcoholism and drug addiction.
And how long do you think, if everything goes well and it goes as hypothesized, how long do you think it would be until this could be given? I mean, since it's already FDA approved.
Yeah, I think it's not too far away. I think we're probably talking within the next 3 years or so. And let me, so I highlighted a satiety factor, GLP-1, that sort of meant to tell the brain, hey, you've had enough food, slow down. We also have signals from our gut that tell our brain, hey, it's been a while since you've eaten. Why don't you go ahead and eat? A hormone called ghrelin is released from our stomach and acts in the brain to promote feeding behavior. But in individuals that are addicted to drugs, that ghrelin signal can actually cause relapse. So drugs in development that block ghrelin receptors are also being pursued for therapy, not just for the treatment of obesity, but for the treatment of drug addiction.
That's really interesting. Emily, you had a question?
Yeah. Hi, I'm Emily.
Hi.
I was wondering, have you looked at it in the reverse? So let's say there's a habit that you're trying to form rather than break towards something that isn't pleasurable. And so, for example, I forget what that product was on the market, but you like insert it. Yes.
A product that I talked about?
That one.
Yeah.
Yeah.
Right.
So if there was the opposite and you like spiked it, let's say someone hated working out, would that have the equal and opposite effect?
Yeah. It's interesting. So you can, if we focus on non-humans for just a moment, if you excite dopamine neurons, you can shape an animal to do just about anything. So people have shown that animals will actually press a lever just to activate their dopamine neurons. And so could you take this then to humans and sort of give people the oomph to work out if you hate working out? I don't know how close we are to that approach, but I think that if you were able to selectively activate those dopamine neurons in humans, absolutely it would work. The question is, would it work too well? But let's talk just a little bit about depression for a moment. Some avenues of treatment for depression include activating a peripheral nerve called the vagus. And over the past maybe 2 years, we're learning that the vagus nerve, that has therapeutic value for overcoming depression, feeds into the dopamine system. So here's yet another avenue where I like the way you're thinking. We might. I don't think you want to activate the dopamine neurons hard directly, but sort of indirectly in moderation.
Somehow shift the neurological connection between whatever that is and finding it pleasurable so that that dopamine can spike.
That's right. That's right.
Cool. Thank you.
Sure.
That was a terrific question, Emily. Thanks for asking.
I'm a transformational habit changer, basically, for a living. So I do what you're doing without the super nerdy details, but I'm super nerdy about the details.
Well, let me add just a little nerd level to the details here. And actually, in response to Terry's question, I mentioned the dopamine neurons from the ventral tegmental area to the nucleus accumbens versus the substantia nigra to the striatum. And actually, as habits develop, the idea is that the dopamine neurons the dopamine release starts to go from the nucleus accumbens to the striatum. So when you sit on the couch and open up a bag of chips and it's just one after the other, and you don't even think about it, it's almost that the striatum is taking over. The dopamine activity in the striatum is playing a role in the formation and the maintenance of habits.
That's so cool. Is there a prize where I get to talk to you for an hour if we enter something?
I'm certainly happy to. I don't want to hold everyone else hostage, but
not now.
I'm
but
always
they can join if they want later.
I'm always happy to talk about this stuff.
Thank you.
Is there anyone else that had a question for Mitch?
Hey, Mitch, it's Dave Purcell. I want to ask just one more question. Can you take us through what it is that you want to do? Like, you got the 40,000 seed grant, then like 2.7 million, and you're continuing to do obviously successful research. What's your goal? Do you want to eventually get a commercial product with like a pharmaceutical company, or what ultimately, as you go down this path and continue to look at this, what are you hoping to do? Like if we 5 years from now, 10 years from now, what would be your dream of success?
Yeah, great question. And I'm going to give you an unfortunate answer because I don't really aspire to make drugs that could be put on the market. I consider myself a basic scientist. I'm interested in unraveling the circuitry and the mechanisms involved in reward and reinforcement. And my hope, what I think of as success, is that the people who do the translational work, who bring the therapeutics to humans, I inform them with my work, and then they bring the products to market. So what I think of as success is when the people at pharmaceutical companies see my work and say, yeah, that's a viable target. That's a target that we should be investing in. That's a drug that we should be bringing up for market. I'd be probably a lot wealthier if I made the leap to developing products and bringing them to market myself, but that's just not me.
That was a great question too. I mean, I always think about, our scientists that we fund, they're building a foundation that's going to be built upon by other scientists, translational scientists, and eventually, hopefully, it will go to pharmaceutical companies and there will be new treatments and eventual cures. So I think that was really well said. Great question, Dave. And then if there are no other questions, I think we're going to let Mitch go eat. And I want to say thank you again so much for doing this and giving a talk, Mitch, and thanks everyone for joining.